Welcome to video 3 on the regulation of gene expression. In this video, we're specifically going to talk about the role of transcription factors and how they recruit RNA polymerase to the promoter to activate transcription. So let's start out with the basic structure and function of a transcription factor. Ones that we're going to be talking about are specific transcription factors that are involved in activating or regulating specific genes. These are different from the general transcription factors, which are found associated with every single promoter. In this case here, we're looking at a transcription factor known as MyoD. In the red or orange here, you can see the DNA, and so I'm going to model this in the middle as well. MyoD itself contains these alpha helices that are shown in purple, and it's kind of straddling the DNA, or sometimes we describe this like scissors. MyoD, like other transcription factors, contain two domains that are known as the DNA binding domain and the activation domain. In these images here, the DNA binding domain is the region that's bound to the DNA. And the activation domain in this case is actually this little tail on the end of MyoD. You'll notice that this particular transcription factor binds to the major groove of DNA. And as we talked about in the past, this is the region of DNA that's more accessible. So those nucleotides, the A, G's, T's, and C base pairs, are interacting directly with the amino acids in the protein. Now, when we take a look at this model and the concept of the full gene and the control elements where this particular transcription factor would be binding, what we find is that this transcription factor is going to recruit general transcription factors to this particular region. Those general transcription factors are going to recruit the RNA polymerase to the promoter, and this is going to initiate transcription at the transcriptional start site. So here we're looking at a different version of the same model. This is known as a space film model of the protein MyoD bound to DNA. Like I showed you before, the DNA here is in this orange-red color. The DNA binding domain of MyoD, and this is going to bind to a specific control element, is shown here, and then at the end of this is our activation domain, and this is the region that's going to recruit those general transcription factors and ultimately the RNA polymerase to the site where transcription will be initiated. Now, I've used this term control element a couple of times to describe where MyoD is binding. The thing to note here is that every single specific transcription factor binds specifically to a specific sequence of DNA that we call a control element. And for each of these transcription factors, this control element may be a different sequence. So for example, in the case of MyoD, here is where the control element is going to be located. There's actually going to be one on the other face of the DNA as well, that we cannot see in this image. And to the right, what you're looking at is known as a web logo. And this shows us essentially the likelihood that we're going to find different bases at different locations. In this particular web logo, height is correlated to the frequency or the likelihood that we will see that nucleotide at that position. In cases where we have smaller peaks, for example, like the first two nucleotides that are A stacked on top of G, the smaller height of that overall particular nucleotide suggests that it's an A or a G, but we have less certainty as to which it's going to be. So if we take this web logo here, we can convert it into what we call a consensus sequence. And this is our best guess as to the sequence that MyoD binds. You'll see here that I've written NN for the first two nucleotides, and this means that it could be anything, but based on the web logo, we know that it's likely an A or a G. The next, CAGC, and I'm choosing C here because it seems to be more frequent than the G and A below it. TG, and then finally N, because that peak is so small, indicating that it could be any of the 4 bases. All right, so just to review this process again, the MyoD, or the specific transcription factor that we're discussing, is going to bind to a control element site-specifically, meaning that it binds to a specific DNA sequence. The activation domain then is going to recruit general transcription factors, which are shown here in purple. Those general transcription factors help to recruit the RNA polymerase, which will bind to the promoter and is placed directly over the transcriptional start site to begin transcription of the mRNA. Transcription and transcription factors are involved throughout the life of a cell, but one of the things that we need to note here is that this process of which genes are turned on and which ones are turned off is really important to what we call cell determination and cell differentiation. Cell determination is really what is a cell going to become, and in particular we're talking here about stem cells. Stem cells are cells that are thought to be able to differentiate into any other cell type. And differentiation is the process by which a cell reaches its final fate. What I'm showing you here is a breakdown of hematopoietic stem cells, which are ones that become different types of red blood cells and white blood cells. You can see here in this image that the multipotent hematopoietic stem cell, or HSC, can, based on the signals it receives, become a common myeloid progenitor or a lymphoid progenitor. And based on the signals that they see, they will become many different cell types. What we're looking at here in this image are the different growth factors or stimulatory factors that multipotent hematopoietic stem cells are needed to be exposed to become different cell types. You may recall we've talked about growth factors in the past and how they are involved in activating signal transduction pathways, and that the activation of those signal transduction pathways often results in a change of gene expression inside of a cell. I want to point out a couple of things here in this image. First, take a look at these two over here, from the common myeloid progenitor to megakaryocytes and erythrocytes. Note that these two are exposed to very similar factors, SCF, IL-3, and GM-CSF. However, the difference of a fourth factor, TPO or EPO, determines whether these cells become megakaryocytes or erythrocytes. The other thing I want to point out is that it's not only which factors are these particular cells exposed to, but it's when they're exposed to them. For example, both HSCs and small lymphocytes are exposed to IL-1, but it's the combination of IL-1 plus the additional growth factors and when they are exposed to them that lead to HSCs becoming common myeloid progenitors or small lymphocytes developing into T lymphocytes. So let's go back to something that we've talked in the past about, and that is the signal transduction pathway. And let's put this all together. We've talked about how cells with cell receptors can, depending on the cell receptors that they have and depending on the factors that they are exposed to, can activate signal transduction pathways. These signal transduction pathways can regulate proteins. One of those forms of gene regulation that we've talked about, for example, they can trigger a phosphorylation cascade. And that phosphorylation can lead to a change in localization of a protein, for example, from the cytoplasm into the nucleus. And within the nucleus, these proteins can bind to DNA, recruit general transcription factors, and recruit the RNA polymerase to activate transcription. So based on which receptors a cell contains and based on the growth factors it is going to be exposed to, that can affect gene expression by regulating which set of genes are going to be turned on or turned off at any given point. My hope is that with this example here, you're able to start seeing how the different ideas that we've talked about throughout the year all connect together. gene expression is not an isolated incident. Instead, it's part of a larger cellular phenomena.